So how did my work evolve? I have to say, when I got to Kevin Egan's lab, it was really a tough time for me. Because a week after I joined, everyone in the lab went on a winter vacation, and I was left alone in the lab to try to figure things out. It was very challenging because the work in Kevin Egan's lab, that is, making pluripotent stem cells ethically, involved a lot of complicated techniques I hadn't learned before, like how to reprogram cells and how to culture embryonic stem cells. I worked very long hours in the lab to make sure that I could master these techniques, and I helped carry on the group's projects as they were on winter vacation, and when they got back, they were very impressed by the results and had a lot of confidence in me. When the group got back, I got to meet key members of Justin's team. There was, of course, Justin Achita, who was the postdoctoral fellow in Dr. Egan's lab that was kind of orchestrating everything. Then there were also two graduate students in Dr. Egan's lab, Esther Sun and Julia Chung. However, when everyone got back from break, I was really interested to learn that Justin's work was really a collaboration between two laboratories, between Dr. Kevin Egan's lab and also Dr. Lee Rubin's lab. Dr. Egan's lab is really an established expert in embryonic stem cell biology, but Dr. Rubin's lab is really an expert in the screening of chemical libraries. So this collaboration between Dr. Egan's lab and Dr. Rubin's lab allowed for the screening of chemical compounds that could reprogram cells to stem cells. I was very much encouraged by this collaborative atmosphere. However, when everyone got back, I learned from Justin that really nobody in Dr. Egan's lab had any experience with chemical screening. So Justin asked me to go down to Dr. Lee Rubin's lab to learn chemical screening techniques and to get the chemical screens going. Chemical screening is a very, very difficult technique. It involves the screening of thousands and thousands of chemicals in order to find the right chemical that you want. It's a really time-intensive technique and is very rigorous, and it also requires the advanced usage of robotics. However, when I got down to Lee Rubin's lab to learn the chemical screening techniques, I was really, really excited because chemical screening is a very powerful tool. It's used by pharmaceutical companies to try to find drugs, and now I would be repurposing it to try to find chemicals to ethically produce pluripotent stem cells. When I got to Lee Rubin's lab, I really had the tutelage of very experienced people in screening, such as Dr. Calvin Lamb, the Harvard Director of High Throughput Screening, as well as Anthony Arvanites, the Harvard Manager of Chemical Informatics, and also Joel Blanchard, who was an assistant in the laboratory. So, after I learned these chemical screening techniques from them, I worked to bridge the gap between Dr. Egan's lab, which was good at stem cell biology, and also Dr. Lee Rubin's lab, which was good at chemical screening. Things, I have to say, did start out slow. I immediately wanted to reprogram human cells to human stem cells that would have direct clinical application, but this was hard because of a lack of technical tools. So, we all decided to start working on mouse cells to try to reprogram mouse cells to mouse stem cells. I'm really excited to say that I achieved excellent results with this. It's known that you need 4 viruses to turn mouse skin cells into mouse stem cells. However, the mouse stem cells you get are obviously really compromised because they've been infected with viruses and contain pro-viral integrations in their genomes that seriously compromise them. So, I did a series of chemical screens, and I found that a single chemical compound can replace 2 of the viruses required in the reprogramming of mouse cells to mouse stem cells. Therefore, I've identified a single chemical that halves the number of viruses needed to make mouse stem cells, and this is really important in our goal of trying to make stem cells without viruses. So after I got my success in reprogramming mouse cells, I started as fast as I could to try to reprogram mouse cells into human cells into human stem cells. Initially, there were lots of technical problems. I tried for months and months to try to find chemicals that could turn human cells into human stem cells, and I was really unsuccessful. I tried a lot of crazy ideas, and none of them worked. None of us really knew what the problem was. So I went out on a limb and took a very creative and unconventional approach called a luciferase-based reporter screen to try to find chemicals that can replace viruses required for the reprogramming of human cells to human stem cells. And after screening about 8,000 chemical compounds, which is the largest that anyone has ever screened at the Harvard Therapeutics Screening Center, I'm glad to say that I have identified several chemicals that may help to reprogram human cells into human stem cells. My results are very important because they show that we can ethically make human stem cells for regenerative medicine to treat human patients. So where does all my work fit in? In conclusion, my work has shown that chemical compounds can be used to ethically create pluripotent stem cells for cell therapies that will be useful in treating many human patients. A big problem with pluripotent stem cell therapies right now are that human embryonic stem cells are normally made from destroyed human embryos. My work shows that you can just turn normal cultured cells into stem cells, thus cutting out the need for embryos and circumventing all these ethical concerns. I feel that my results are very important for advancing the field of regenerative medicine with human patients. In my work, I've really been influenced by the ideas of Dr. Shinya Namanaka in Japan that really had the first ideas that you could directly turn any cells into pluripotent stem cells without using the embryo. I've also been very much influenced by the ideas of Brent Stockwell, who had the idea that chemical compounds can really be used to manipulate specific signaling pathways in human cells. This helped give me the idea that chemical compounds could potentially be used to manipulate cell signaling pathways in skin cells that would be able to turn them into pluripotent stem cells. I'm really proud to say that I'm a Davidson Young Scholar, and if I did get the Davidson Fellows Scholarship, I would be glad in telling other people how much the Davidson Fellows Scholarship has enabled me to go to Stanford for graduate school, and how it really helped me, as a 16-year-old graduate student and scientist, to really pursue my dreams of a scientific career. Thank you very much for your consideration. It means a lot to me. Please feel free to contact me at any time if you have any questions.